Welcome to Chapter 8 of Microbiology. This is the last chapter of the Unit 2 section, and we're going to talk all about genetics of microbes. A lot of this chapter is going to be very definition heavy, and then we have kind of roughly three big processes. So the last two are technically two parts of a bigger process. So let's go ahead and jump in. To get us started with genetics, we need to talk about this term heredity. Heredity is going to be this process by which traits are going to be passed from parent to offspring. And I want you to imagine that there are two possibilities of passing traits from parent to offspring because it's not always just parents. It can be a single parent. So when it comes to that transmission of traits, it can be the combination between two parents to an offspring. That will be that process of sexual reproduction, where you have two parents with their gametes mixing, producing that offspring, as opposed to just one parent asexually reproducing and producing a identical clone of themselves in that offspring. However, with that asexual reproduction, we don't usually get a whole lot of variety or genetic diversity within that type of population, as opposed to organisms that sexually reproduce. We do get a lot of genetic diversity, but you still see mutations between the two types of reproduction. Now, when it comes to microorganisms like bacteria, we do have a little bit of a weird thing to throw in there, and that is something we've already talked about, and that is horizontal gene transfer. So we will talk about that again in this chapter coming up near the end. So kind of keep that in mind because that throws things off a little bit with that asexual reproduction because it's not always so kind of clean and precise as far as one parent producing just a clone of itself. We also have that horizontal gene transfer happening as well. So let's talk about some more terminology. We have these first three terms, and I want you to know that it has a lot to do with how much DNA it involves. So for genome, this is the total amount of DNA within an organism. So the whole thing. For eukaryotes, this is going to include all of the DNA within every single nucleus, as well as the DNA within the mitochondria, as well as if we're talking about plant cells, the chloroplast, because remember mitochondria and chloroplast have their own individual DNA as well. So that's a lot of DNA to take into account. A lot. That is the genome. For prokaryotes, it's actually a really small amount because these are single-celled organisms, and it's just that one single chromosome found in the nucleoid, which is that region that we find that bacterial DNA, as well as whatever that little extra pieces of DNA might be, which are called plasmids. So for prokaryotes, that genome is going to be substantially smaller than in eukaryotes. So a large amount of DNA, meaning the genome. Okay. With chromosomes, these are going to be pieces of that DNA. So if the genome is like an entire textbook, chromosomes are going to be like chapters within the textbook. For humans, our chromosomes are going to be in total 46 pieces. They are also linear, as opposed to a bacterial cell, which is a single chromosome and it's circular. So kind of keep those differences in mind. From there, we have this term gene. Genes are going to be segments of DNA on a chromosome. That is very important. So they are segments of DNA on a chromosome. So again, if a genome is going to be our textbook, chromosomes are going to be the chapters within the textbook. Genes are going to be the specific pages within the chapters. So kind of keep that in mind as far as how those terms go together. Again, it's having to do with the amount of DNA that we're talking about with those terms. Then at the bottom we have these two terms, genotype and phenotype. The genotype is going to be the genetic coding or genetic makeup of an organism that is going to code for a specific trait. The phenotype is that trait itself. A phenotype is the physical manifestation of that genetic coding. So those are those two differences there. Kind of keep that in mind. Now, just to give you kind of a better idea of those differences between genomes for a prokaryote and a eukaryote, I do have this slide where we're looking at the genome of E. coli versus a human genome. So what we have going on is that the E. coli has, as we would expect, a single chromosome containing roughly 4,000 genes, as opposed to the human genome, which is going to have 46 chromosomes and 20,000 genes on those 46 chromosomes. And that's actually probably a lowball estimate. So humans have about 5 times more genes than an E. coli, and that makes so much sense because those 4,000 genes, very small amount, is going to create what is a single-celled organism, as opposed to 20,000 genes that is going to code for an entire multicellular differentiated celled organism such as a human being. So it makes sense that our genome has to be so much longer, so much bigger than something that is single-celled. We keep talking about DNA, so let's talk about what makes up DNA. We have nucleotides, that is going to be the building blocks of DNA, but we want to talk about what nucleotides are made of. So what are the building blocks made of? Well, we're always going to have a sugar, a phosphate group, and a nitrogenous base. So those are the three important pieces of a nucleotide, which is going to make up DNA. But what I need you to kind of point out on this slide to yourself is that there's some differences between DNA and what we're going to talk about eventually, and that is RNA. So DNA is going to be deoxyribose, which is going to be the type of sugar that we find in its nucleotide. For RNA, it's going to be ribose. The phosphate group is still going to be the same, but the nitrogenous base is going to be different. So the nitrogenous bases that are going to be found in DNA is A, T, C, or G. So adenine, thymine, cytosine, and guanine. Now, most of those are the same when it comes to RNA, except for thymine, the T. Instead of thymine, it becomes uracil, which is going to be a U. That is the two big differences in the nucleotides of DNA and RNA. And we're going to kind of keep coming back to that over and over. This is a great first slide to make sure that you start to understand that and start to remember this. So this slide is going to start to introduce us into how we put those three parts of the nucleotide together and how they stick together, how they stay together, and what order they stay together, and what direction we're going to read the nucleotides together. Now, that is to say, this slide is all text. On the next slide, we have an image, and that is going to be much more helpful. But for now, just bear with me as we get through the text, and then we are going to get to that next slide, and I'm going to talk through it again when we have a visual. So an important thing to keep in mind is that DNA is going to look like a ladder. And I'm sure you kind of have an image of this already, that DNA looks like a double helix. It looks like a ladder that's been twisted. When we look at this ladder, it has two important parts. It's going to be the backbones, that is the sides of the ladder, and then the rungs, the part of the ladder that goes across the middle. So those backbones of the ladder are going to be alternating chains of sugars and phosphates. Now, what's interesting about the backbones that have those alternating sugar and phosphates is that one side, one backbone, is going to be read in a particular way, while the other side is going to essentially be the opposite. And you're going to see that better on that next slide. Now, how we properly read this DNA, these sides, is going to be from 5' to 3'. All that means is we're reading from a 5' carbon to a 3' carbon, and that's all you really need to know. But I would highlight or write or put a star next to something that says near this slide that we are reading from 5' to 3' forever and always. That is an important thing to make sure you understand. So one side, one backbone of our ladder is going to be 5' to 3', and then the other side is going to be the opposite, where it's going to be 3 to 5. And that's very important. The middle part, those rungs, are going to be our bases, those nitrogenous bases, and they're going to alternate as well based on some pairing rules, which we'll talk about in a minute. So let's go to that image and then talk about this again. All right, here we have that image of our ladder. So here and here are going to be those backbones, right? So our backbones being the sides of the ladder, right? And then that middle portion, okay, which I'm going to indicate with also right there in the middle, is going to be our rungs. So we have our backbones on the side, which is alternating sugar phosphates, and the middle there is going to be the rungs. So on one side, we're reading from 5' to 3', and I forgot my little prime there, but we have 5' to 3', which is going to indicate on the other side, we're reading from 3, and I'm sorry, that's so ugly, I'm doing this with a mouse, to 5'. Okay, so they're going opposite. Okay, we've got our opposite directions. Okay, they are inverses of each other. What happens with the bases here is very important. We've got our G to our C, T to A. Now, what's really important to remember when it comes to DNA is that this entire side is one strand, meaning that, if I could get another color, this entire side is the other strand, and together they make that double-stranded DNA, right? So that's a very important thing to make a note of. So we were just talking about our 5' to 3', that is how we read, right? So 5 to 3. Again, I'm so sorry, that's so ugly. Ooh, extra ugly. So 5 to 3', very, very important. That is the direction that we read. We've got our phosphate sugar backbones, and then our rungs made up of the nitrogenous bases. And then one side of the DNA is going to read in one direction, and then the opposite side is going to essentially be like the inverse with that matching nitrogenous base. So let's talk about those nitrogenous bases next and see what those pairing rules are and how these things stay connected. So I'm going to kind of jump back and forth on this slide. We have our pairing rules at the bottom. That is our bases, how they pair together. With DNA, it is adenine to thymine and thymine to adenine. With guanine, that is pairing to cytosine, and cytosine to guanine. Now, what's important to note is with RNA, guanine and cytosine also go together, cytosine and guanine, but adenine will not pair with thymine, it will pair with uracil. There is no thymine in RNA. Very important thing to make a note of. However, thymine, going from DNA to RNA, is going to still pair with adenine. And we're going to see kind of examples of this in a little bit. Now, how these sugars and phosphates, as well as our nitrogenous bases, stay together is going to be through bonds. When it comes to the sugar and phosphate groups, they are going to be very strongly bonded together through covalent bonds. This is a very strong bond, so that is through the sugar phosphate groups. Now, the nitrogenous bases are going to have a weak bond because that is going to allow them to be unzipped. This is going to be through a weak bond called a hydrogen bond. This allows them to break apart, which is what we can see happening in that little animation happening there. We can see that the sugar phosphate groups are not breaking apart because they have such a strong bond. But the hydrogen bonds, that is what is breaking apart so that we can rebuild them when we're replicating or making RNA or whatever it is that we're creating. But that is what is happening. So maybe you're asking this and maybe you're not. Maybe you're just trying to get through this lecture, and that's okay too. But maybe you're asking, why does guanine have to pair with cytosine, and thus cytosine have to pair with guanine, and adenine to thymine, and so on and so forth? Well, it has to do with this rule being that purines have to pair with pyrimidines. So that is the rule. So the nucleotide bases are going to be one or the other. It's going to be a purine or a pyrimidine, and they always pair with each other. The purines are going to be guanine and adenine, and the pyrimidines are cytosine, thymine, and uracil. So they're kind of like puzzle piece shapes where they have to pair together. Now, what's kind of weird is why can't guanine then pair with thymine? And it has to do, again, with the shape as well as the bond. So we are going to talk about the bonds next. When adenine bonds to thymine, it is a double bond, as well as when adenine bonds with uracil, it's going to be a double bond. That's why those specific nucleotide bases are going to be able to bond with each other. As opposed to cytosine and guanine, they're going to have a triple bond. So when it comes to guanine not being able to pair with thymine, even though guanine is a purine and thymine is a pyrimidine, those can't go together because a double bond cannot go with a triple bond. It essentially won't bind properly. So again, when we talk about these bonds, we're talking about hydrogen bonds. And again, it's still a weak bond, but they're still holding together. And again, that weak bond is going to happen so that they can break apart. This is a really nice image showing us those sugar phosphate groups along the side. Remember that those are being held together with a covalent bond, which are really strong. Those aren't going to break apart. And then we have the hydrogen bonds. We have the double between the thymine and the adenine there. We have a triple bond between the guanine and the cytosine, and then so on and so forth as we move through those nitrogenous bases. showing us those specific hydrogen bonds. And again, those are going to easily break apart when we want to unzip the DNA, when we want to make a replication or go through RNA, whatever it is. What's kind of neat here in this image is that we can see with these three pieces right here, this is going to be a nucleotide, right? So this is the three parts. We have the phosphate, we have the sugar, and then we have the nitrogen base. So those are the three pieces that make up a nucleotide. Remember that those are the three pieces that make up DNA, that nucleotide. I have two more terms for you to know. Granted, the terminology is going to be something that we kind of understand already as far as anti-parallel. This is going to be that concept of going from 5' to 3' on one side of the backbone, and then on the other side of the backbone going the other way, the 3' to 5'. So it's that the backbones are parallel, but in moving opposite directions. So that is anti-parallel. This term semi-conservative is going to refer to how DNA is going to make new DNA. So with conservative methods, that is going to be taking that original double-stranded DNA and not breaking it apart. You're just going to make an entire new piece of, or new pieces, a new double-stranded DNA from scratch. With semi-conservative procedure of making DNA, it is taking the original piece of DNA, breaking it in half, and then copying that half of the DNA and filling it in. So instead of making the DNA completely from scratch with the conservative method, you're taking half of the original piece and just filling it in. This is going to make it to where it's less likely to make mistakes because you have a template to go off of. So if you can see in that image on the right with the semi-conservative, the original pieces of DNA are blue. In the semi-conservative, we are filling it in where we have broken that DNA in half, we have unzipped it, and filled it in with that new piece of DNA in red. So it's kind of a built-in safety measure, and it is something that our DNA does when our cells go through replication. Kind of interesting. It's important to understand that literally every living thing that we know of, and this could change, who knows, but as of right now, what we understand about all living organisms is that they are made of DNA. So that is the building blocks of all living organisms. All living organisms have DNA. And the only real difference is the amount of DNA and then the order of those A's, T's, C's, and G's. So if you want to take it as, I don't know, a species, humans have a lot of variety, but it's just that order of the A's, T's, C's, and G's, but we all roughly have the same amount of DNA. And actually, a lot of that order of A's, T's, C's, and G's is actually pretty similar too, which is going to give us our very similar shapes and the fact that we are human. As opposed to something like, a flamingo, which is going to be something slightly different. It's going to be a bird, something that has feathers, something that has many different functions and features than a human. The amount and then the order of the A's, T's, C's, and G's is going to be very different than ours. They produce different proteins or a different combination of proteins to make their features compared to us. Because remember, DNA, that end-all be-all goal is for DNA to make RNA, which will then make those proteins. At the beginning of this chapter, I said that there were three big processes that we would talk about. The first one is going to be replication. We'll talk about those last two in a little bit. Now, the purpose of replication is going to be to replicate DNA. So this is going to be when a cell is making a new cell. So when a cell goes through cell division, it needs to replicate its DNA in order to create that new cell. In eukaryotic cells, this is going to happen in that process called mitosis. So making a new cell, we're also going to have that happen in meiosis and making gametes. Don't worry about that too much. But for microbes, for bacteria, this is going to be how that cell is going to make its offspring. It's going to replicate its DNA so that that replicated chromosome goes into the new cell. There are 4 important enzymes that we need to know what they do and also the name of that enzyme. The first is helicase. Helicase is going to unzip the DNA. It's going to break apart those hydrogen bonds between the nitrogenous bases. So that's a very important part of helicase. Unzip and break the hydrogen bonds. Or unzip by breaking the hydrogen bonds is a better way to say it. From there, we have DNA polymerase 3. This guy is going to be, and I want you to write it here, the main builder. It's going to build new pieces of DNA on one side of the DNA. It's going to be very fast, but it's also going to be a little like a checker for itself because it might make a mistake. And if it does make a mistake, it doesn't have time to correct itself. It's going to kind of throw a flag on the play. So the enzyme that's going to come in after DNA polymerase 3 is DNA polymerase 1. This guy is going to see all of those mistakes and fix whatever mistakes that DNA polymerase 3 made and checked in on itself. So it's going to be the fixer. DNA polymerase 1 is the fixer, whereas DNA polymerase 3 is the builder and checker. And then we are going to bring in this 4th enzyme. Now this guy's going to make a lot more sense when we talk about how the different backbones are going to be replicated. So remember, those backbones are being read from 3' to 5'. So that's the other way around, ignore me. 5' to 3', 5' to 3', that is how we read them. So in reading those backbones from 5' to 3', this ligase is going to be important on the strand that is the opposite side, that is going from 3 to 5. And we'll see why here in just a minute. The ligase itself is going to be a glue that is going to bind DNA together. So just bear with me for a second and we'll see why this guy's important. Again, remember, we are going to read and synthesize or make DNA 5 to 3', always and forever, meaning that those backbones that we have broken apart, those nitrogen bases, those hydrogen bonds have been broken apart, and now we have 2 strands. Those strands are going to be the leading strand and the lagging strand. The leading strand is going to be, you can kind of think of it as the good one, the perfect one, the one we like. And the lagging strand is going to be the one that's backwards. The leading strand is going to be the one that naturally reads 5 to 3', while the lagging strand is reading backwards 3 to 5'. Now, where this comes into a problem is where that DNA polymerase needs to build that DNA. On the leading strand, that DNA polymerase 3 is going to be just fine. It can build the DNA 5 to 3' in one single move. totally fine. But because DNA polymerase 3 has to synthesize DNA 5 to 3 prime every single time on the lagging strand, it has to go backwards and then move forwards, backwards and then move forwards, therefore making the DNA in segments. So that's kind of an issue. That's where that ligase comes in and it has to glue the pieces together. So let's look at an image that might help us visualize what's happening. Again, remember that DNA is always going to be made 5' to 3'. So what we have happening here at that number 1 spot, let me get my little guy here. We have what's going on, the helicase unzipping, yes, unzipping our DNA, and we have the leading strand and then the lagging strand. The leading strand, we are able to have that DNA polymerase build that copy of DNA in one piece because we are able to build it from 5 to 3 prime, easy peasy, right? Now on the lagging strand here, this one at the bottom, we're having a little bit more of an issue where that DNA polymerase is having to move back and then forward, back and then forward, back and then forward, right? And that is because that polymerase, that DNA polymerase 3, is still having to build from 5' to 3', but it has to go backwards and then forwards and then backwards and then forwards in order to build that DNA. And that's where that ligase is going to come in and start gluing these pieces together eventually once it's done building, in order to glue this entire piece of DNA together. So that's kind of that whole process there. What we have here is an explanation, kind of still what we were just talking about, that DNA polymerase 1 is checking. We also have some primers coming in. Those guys are going to kind of lay the groundwork there so that the DNA polymerase 3 is able to start working and make that DNA copy. Those fragments of DNA that are having to be glued together on the lagging strand are known as Okazaki fragments. That term is just coming from the person who discovered them, which is kind of cool. So just kind of keep that in mind. Again, those Okazaki fragments are having to be glued together because the DNA polymerase 3 is having to move backwards and then forwards and then backwards and forwards because it has to make the fragments from 5' to 3', and that lagging strand is kind of backwards. It is going from 3 to 5, not 5 to 3. And then here we have this lovely little illustration of our helicase coming in and unwinding or unzipping our DNA. We have those primers coming in, getting our leading and lagging strands ready for the DNA polymerase, and they are building those fragments. The leading strand we have being built 5 to 3' in one gigantic piece, while on our lagging strand we have those Okazaki fragments being built because it has to be built 5 to 3', but because that fragment is 3 to 5, it has to go backwards. So hopefully this illustration and animation helps you understand this process. Watch it as many times as you feel is necessary, but understand those differences between the leading strand, the lagging strand, what's happening with the DNA polymerase. We've got those Okazaki fragments. Again, we have to read from 5 to 3, moving backwards on the lagging strand, moving forwards on the leading strand. And then again, that little primer coming in and preparing things for the DNA polymerase to actually build on those strands themselves. In another illustration, just showing the same thing, just kind of a quicker illustration. This one includes the ligase coming in and gluing the Okazaki fragments together on the lagging strand. So this process of DNA replication is kind of easy to think about where the start of a chromosome begins for our chromosomes because they're linear, they're pieces of DNA, right? Versus bacteria, which are circular chromosomes. So where on a circle is the start? Well, there has to be a starting signal, which we call the origin of replication site. It's still going to have to read 5' to 3', but this process of DNA replication is going to be a lot faster because the DNA polymerase can go in both directions, still in 5' to 3', but it is bidirectionally. So it can go in both directions on that circular chromosome. It doesn't have to go forwards and then backwards, forwards and then backwards, forwards and then backwards, that kind of thing. It's able to just kind of start and go around. So one of the big differences between DNA replication in prokaryotes versus eukaryotes is one, bacteria are a lot faster at replicating their DNA, which makes sense because you can have a new generation of E. coli in 20 minutes versus when can humans have a new generation? It takes quite a while before there's an entire new generation of humans compared to like E. coli. And that has everything to do with it's a single cell of E. coli replicating its DNA. It's smaller and it's also only one, has everything to do with that circular DNA being able to replicate bidirectionally. Here's an image of that origin of replication, that starting point on the circular DNA, that replication beginning, so it's forking off of the parent strand, and then there is that termination site where that ending point of making the replication is going to end, and then they break off from each other, and then you have that new piece of DNA essentially. So what's kind of neat is that both strands are a leading strand. There's not a leading and a lagging. It's just that both strands are the leading strand because it is bidirectional. As opposed to in eukaryotes, there's a leading and a lagging strand. So another difference between prokaryotes and eukaryotes when it comes to DNA replication. So now that we've seen how genetic information has passed from a parent to a daughter cell, which is what we call that product of binary fission or mitosis, those are daughter cells. There's no such thing as son cells. Sorry, guys. So that is that product cell, correct? We're going to look a little closer at how genetic information is moved within a cell. And then later we're going to look at how bacterial cells can also transfer genetic information between unrelated cells. Remember, that's horizontal gene transfer. So that is going to come up later, probably in chapter 8, part 2. So we are going to start with that next process. So we talked about replication. That's the first big process. Now we're going to talk about transcription, which is going to be that transformation of DNA into RNA. That is going to have a second part, which is translation. And then together, transcription and translation together is going to be something called gene expression. Okay, so we have transcription, translation, together it's gene expression. Let's start in on gene expression with transcription. This first part transcription is going to serve the purpose of converting DNA into RNA. There are a couple of main players here. We have that DNA template, the RNA polymerase, and then our nucleotides, which is going to be different than the nucleotides we used in DNA, but we still need that DNA template, which is going to be our A's, T's, C's, and G's. But remember, that is going to change with our RNA, which is going to be A, U, C's, and G's. So everything's going to be the same, except thymine is not going to exist in RNA. It's going to be uracil. Very, very important. Now, there are important sites that we're going to find in the DNA, much like in our replication of DNA. We have that origin of replication when it comes to the bacteria and the termination site. It's not dissimilar. We have a promoter and a terminator. So the promoter is going to be where the RNA polymerase, remember that's going to be that builder. When we look at replication, it's not dissimilar here with transcription. Polymerase is still going to be a builder, but it's also going to be the unwinder. So we have RNA polymerase binding at the promoter site, and then the terminator site is going to be where the RNA polymerase is going to release, because that synthesis of RNA will finish at that point. Promoter, starting point, terminator, the ending point. Now here's an image showing us what's going on. I'll try and kind of point out the important parts. So we have that promoter area, so where we're going to start unwinding and reading the DNA to make the RNA. So we have the RNA starting to be made here, and that is that little teal line, and we can see that kind of zoomed in image here where we are making a copy of the DNA, but using the nucleotide, or I'm sorry, the nitrogenous bases of the RNA to make the RNA based off of the DNA. So then we see that RNA getting longer and longer as we are moving down the DNA. So we are starting somewhere here, and then we are going to end somewhere over here, right? So we started over here, we're ending here, and that is where that terminator ends. That RNA polymerase is going to pop off, and that is going to be the end of it, giving us our DNA being rewound, where we just looked at this gene, because then it's just going to look at a segment there, making the RNA. And then we have our segment of RNA here. So our DNA has been rewound, and then we have our RNA strand here. Please keep in mind that the RNA is not going to be bound in any way to the DNA. We are using the DNA as a template to create the RNA. It is an instruction manual to make the RNA, which will then be used as further instructions to make the amino acids that make the protein in translation. Just to recap real quick, we are going through the process of gene expression. In this first part, we are going through transcription. We've gone from a strand of DNA, we have transcribed it, which is to say we've made a copy of it into RNA. So, with this RNA, we want to talk about some of those differences. Remember, we talked about this at the beginning of this chapter. One of the big differences being that it's a different kind of sugar. It is ribose instead of deoxyribose. It's going to contain uracil instead of thymine. And then another big difference is that RNA is single-stranded instead of double-stranded like DNA. So it's just a single strand of RNA. So some big differences here. You should be able to tell on an exam if I'm giving you DNA versus RNA, because DNA will contain thymine. So you'll see T's instead of U's. If there is a U, I am talking about RNA. So kind of keep that in mind. So let's see if we can get this guy going and playing. I know it takes a little bit longer. So here we've got our RNA polymerase coming in, and we see that promoter site. Yep, there we go. So that RNA polymerase is coming in. It's attaching at the promoter site and unwinding the DNA. What we're seeing is that RNA being made, again, 5' to 3'. It's reading the DNA, making a copy into that RNA. Very important. We're still going from 5' to 3'. Still making those connections. We've got our RNA growing. Waiting until we get to our termination site. That RNA is not connected to the DNA at all. Once it hits that termination site, that RNA polymerase is going to detach, and we have our DNA rewinding, and then that RNA strand has been made. Now I have kind of sort of been lying to you, and I'm sorry. It's not a big lie. It's more so like what specifically we should call the RNA that is made from transcription, and that is mRNA. It's messenger RNA. This is going to be the RNA that is produced from transcription, and it's going to be what encodes a protein, and it's kind of the message from DNA, if you want to think of it. It's sort of the middleman. There are two other types of RNA molecules as well. We have ribosomal RNA, or rRNA, which is going to be part of the ribosome that is going to help create the amino acid chain that will create the protein in transcription. And then we also have the tRNA, or transfer RNA, which is going to carry the amino acid to the growing protein chain, which we'll talk about this in the next section. So kind of hold on to these terms here, and I'll see you for Chapter 8, Part 2.